So hello everyone. Today I am going to talk about a recently published article about structural and functional study of rotavirus capping enzyme VP3. So rotavirus, as we know, is a major agent of severe acute diarrhea among infants and children. Despite having vaccines available, approximately 200,000 kids die each year due to rotavirus infections. While these vaccines are very effective in the developed world, the efficacy of these vaccines are compromised in lower income countries like India, some other Asian countries, and African countries. So, and due to this low efficacy, we need to think about some alternative antiviral strategies so that we can tackle this virus more effectively. And this is where our recent article could be very helpful. So if we look into this rotavirus structure, it forms a triple layered particle like structure and it contains segmented double stranded RNA genome. Now this infectious triple layered particle like structure is formed by 6 different viral proteins. Now there are 3 layers formed by 3 proteins. Then we have one spike protein, and inside the particle we have 2 more proteins. One is rotavirus RNA dependent RNA polymerase VP1, which makes copies of viral RNA genome, and then we have another protein that is viral capping enzyme VP3, which puts a cap on nascent transcript and help in the efficient translation of viral RNA viral proteins. Now this is all is known about this virus, and our lab has been working in the rotavirus field since more than 3 decades in close collaboration with Professor Mary Estes lab at Baylor College of Medicine. And when I joined this lab around 4 years back, we had structural and functional information available about 5 of the viral proteins from the 6 of this whole rotavirus particle. Now the only protein for which we had a very limited understanding in terms of function and structure was a viral capping enzyme VP3, and this is where we started looking into. Now, with using a combinatorial structural biology technique like X-ray crystallography and cryo-electron microscopy, we have been able to determine a high-resolution cryo-EM structure of this capping enzyme VP3, and we also validated that structure by X-ray crystallography structure. So, what we found in our study was one of the most fascinating part of the study was the oligomeric or high oligomeric structure of this protein. So when we look the micrograph and when we look the pattern of gel filtration column, we could clearly see that this protein is forming a higher tetrameric assembly, which is very stable in different pH ranges. And when we demarcate different enzymatic domains in that tetrameric assembly, we could very clearly see that this assembly would make this enzymatic machinery or this capping machinery very efficient. So this assembly can perform multiple capping activities simultaneously. Now, in addition to that, we also did some very interesting biochemical studies, and we were able to first time show that perform that this protein can perform an RNA triphosphatase or first step the RNA capping activity, and we could demarcate that which domain is doing that activity. And in addition to that, one of the interesting finding of our study was that what happens when there is a transcription going on in the rotavirus. What happens is there is a transient duplex formation between the nascent transcript and the template strand. Now, when this duplex comes out from the exit tunnel of RNA polymerase, this duplex has to be separated before the capping process. So it was not known that which enzyme or which protein might be doing this function. Since we have established that this protein has an RNA helicase activity, we believe that this protein also might be doing this separation part of the nascent transcript with the template strand followed by the capping activity. So this is in brief that we have done in this paper. This is the brief just description. Now this paper also opens up multiple other fronts for us to look into, that what are the things that we can explore in future. One of the most interesting part that I would personally be interested to look into, that what is the oligomeric organization of this protein inside the particle when this transcription is going on. So on the contrary to few other viruses, like if you see just coronavirus, what happens in rotavirus is that when rotavirus insert comes enters inside the human cell, so, through the endosome process. Now inside the endosome there is a lack of calcium ion concentration, which helps in disruption of outer layer of this virus particle, and the release of a double layered particle like structure which has just two layer and the whole viral genome. Now this double layered particle is the transcriptionally active form of the virus. Now if you give all the reagents that is required for the transcription, this virus double layered particle start performing the transcription inside the particle. So it has endogenous transcription, unlike the coronavirus which uses host machinery also, and which performs the transcription in the cytosol. The transcription in case of rotaviruses takes place inside the particle. So now we are interested to look into that, what is the oligomeric organization of VP3 inside the particle when the endogenous transcription is going on. So we want to do this study as a next target, and this is all that I would like to say for this paper, this research study currently. Thank you very much for your time and patience.